The brain in the central nervous system is a sensitive and delicate organ susceptible to numerous diseases. In addition to genetic illnesses, many microorganisms, including unicellular parasites, are attracted to brain cells, causing various diseases. Today, we will discuss parasitic diseases. A parasite is any living organism that obtains nutrients necessary for its survival from other organisms, known as hosts. These hosts also include humans who, when invaded by parasites, may experience neurological, cognitive, or mental health problems related to the central nervous system, including the brain. Upon entering the brain, these parasites release substances that disturb the host's neural circuit. Nowadays, the parasite Naegleria fowleri is a tiny yet formidable creature under debate and has been the center of attraction for researchers. Let's explore the mysteries of this brain-eating amoeba. What is a Naegleria fowleri? Naegleria is a microscopic, free-living amoeba commonly inhabiting soil and freshwater bodies such as lakes, rivers, and hot springs. It can also be found in the host, specifically their central nervous system. It is thermophilic and can tolerate 46 OC or higher temperatures, hence can easily survive in warm water temperatures ranging from 32 to 43 OC. It has several species, but only Naegleria fowleri can cause human infections. How does Naegleria fowleri infect the human body? As Naegleria fowleri lives in contaminated freshwater bodies such as hot springs, rivers, and even swimming pools, it may transmit to humans through the nasal passage. This often occurs when individuals swim, dive, or submerge their heads in fresh water, such as lakes and rivers. Naegleria fowleri infections may also occur when people use contaminated tap water to clear their sinuses or wipe their nostrils during religious activities, sending water up the nose. People have infrequently contracted Naegleria fowleri infections from recreational water that lacked sufficient chlorine, such as pools, splash pads, and surf parks. The amoeba then reaches the brain by following the nasal route, where it degrades brain tissue and causes the lethal disease called primary amoebic meningoencephalitis, a deadly infection. That's why this amoeba is infamously known as a brain-eating amoeba. Primary amoebic meningoencephalitis, PAM, is a lethal condition with a 98% mortality rate. This condition occurs when brain coverings or layers called meninges are infected by Naegleria fowleri. The most prominent feature of primary amoebic meningoencephalitis is swelling in the brain due to meninges inflammation. Immunocompromised patients are more vulnerable to catching the infection. Symptoms. There are no clinical symptoms associated with PAM. General symptoms appear within 24 hours post-infection. However, they may sometimes appear within 5 to 7 days of infection. Patients experience severe symptoms at the start of infection, leading to death 18 days later. Symptoms such as intense fever with painful episodes of headache, vomiting, shivering, neck stiffness, and photophobia or sensitivity to light may result in a coma and, eventually, the patient's death. Diagnosis. Serologic testing. The measurement of antibody titers in infected patient sera against Naegleria fowleri is accomplished by using an indirect immunofluorescent antibody. On the other hand, this information is not particularly relevant given that most individuals with PAM pass away before an immune response can be generated. Polymerase chain reaction, in short PCR. Doctors recommend PCR to detect Naegleria fowleri in the DNA extracted from the cerebrospinal fluid or CSF and tissue samples. The amplification of DNA from cerebrospinal fluid and fresh tissue samples is successful. More and more PCR-based methodologies, such as conventional and real-time PCR, have been reported for detecting and identifying free-living amoebic infections in clinical specimens. However, these methods are currently only accessible at a few number of specialized reference diagnostic laboratories. Centers for Disease Control and Prevention, CDC, is America's national public health agency ensuring the public is healthy, safe, and secure from mild to complicated diseases. After an event of 154 out of 157 deaths caused by Naegleria fowleri infection in the general public of America, the researchers at CDC put a great effort towards controlling this infection. They did groundbreaking research on this life-threatening amoeba and found a novel RT-PCR-based, qualitative method for its detection in clinical samples. Lumbar puncture. A lumbar puncture, often called a spinal tap, is a medical test to diagnose specific CNS-related medical conditions. During this procedure, The doctor collects CSF fluid from the spinal canal of patients infected with Naegleria fowleri. To start, surgeons use either local or general anesthesia to numb the back area. Then, they carefully insert a thin, long needle into the space between two neighboring vertebrae to extract the CSF fluid. This fluid surrounds the brain and the spinal cord and protects them from injury by providing cushioning. Your healthcare provider may recommend lumbar puncture or spinal tap if you have symptoms like hallucinations, vomiting, headache, and lower back pain. This particular test is frequently employed to identify the existence of Naegleria fowleri in the CSF fluid. Likewise, patients who undergo lumbar puncture might encounter headaches or discomfort in the back. Biopsy. In this diagnostic procedure, tissues, cells, or fluids from the infected area of the body are removed for microscopic detection of amoeba. Treatment. Medicines such as amphotericin B have amoebicidal characteristics and are strongly recommended for primary amoebic meningoencephalitis. At the same time, rifampin and miltefosine are also the drugs of choice. Fluconazole, combined with amphotericin B, is also suggested in treating some Naegleria fowleri infections. Besides treatment with medicines, keeping the body at a temperature below normal can reduce brain swelling. However, early diagnosis and treating the patient with advised drugs can save the patient's life. Preventive measures for Naegleria fowleri infections. Do not swim in water suspected to have Naegleria fowleri or water that is unclean and still for longer. Always use nose plugs whenever going inside water. Do not use water straight from the tape to clean your nose or mouth. Use distilled or sterilized water or at least boiled water for this purpose. Always use filtered water. Filters labeled NSF 53 or NSF 58 can be used for water filtration. Disinfecting the water is crucial to prevent infection. Always use chlorine or bleach to disinfect water used for nasal cleaning. If you enjoyed this video, don't forget to like, share, and subscribe to our channel for more captivating content. And as always, stay curious and keep exploring.